Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show, bringing another really fascinating guest today involved in creating a better tomorrow. Today, we have the honor to be joined by Professor Noga Kronfeld-Schor, who is a principal investigator and chair in the Department of Zoology, Tel Aviv University, where her research centers on environmental physiology with a focus on biologic rhythms of diverse vertebrate species, ultimately asking questions regarding the consequences and significance of these various rhythms, as well as the interaction between physiological and endocrinological processes, as well as the evolutionary ecology of various organisms and communities. She's also involved in studying the impact of events like global warming, light pollution on biodiversity, as well as on human and environmental health. In addition to those roles, Professor Kronfeld-Schor also serves as the chief scientist of the Israeli Ministry of Environmental Protection. There, she's involved in providing strategic, scientific, and technological consultation to the ministry, promoting the environmental field in Israel in accordance with various advancements being made in leading countries around the world. Professor Kronfeld-Schor has an undergraduate degree in biology, a master's in zoology, a Ph.D. in zoology, all from Tel Aviv University. She went on to do post-doc work at Boston University in zoology and neuroendocrinology. In addition to all those roles, she's also served as a member of the board of directors for the Society for Protection of Nature, was a member of the plenum and head of the scientific and professional committee of the Nature and Parks Authority, and has managed numerous national and international projects along the way. Honored to have with us a lot of really interesting themes to get into today. Professor Noga Kronfeld-Schor, thank you so much for taking the time to come on the show.
Thank you for inviting me. I'm happy to be here.
I'm very happy you're here. A lot of very unique topics that I've been looking forward to getting into. Would love to start things off, as we typically do, by handing you the floor for a little bit, just to talk a little bit more about your background. If you could take us back a little bit to sort of why biology, why zoology, and a little bit of, I noticed but your master's and your PhD both focused on interesting species that like dry, hot, rocky environments, but also have some biologic differences between the Cape hare and the spiny mice. Love to hear about how you got focused and interested on these particular species, and then we'll head down the road to chronobiology in a little bit.
Okay, so actually, as far as I can remember myself, I was interested in science and especially in biology. And I remember as a child asking myself, how come I can't breathe water while some species can? Or how come I can't fly or breathe on high mountains? And these questions fascinated me as a child. I wasn't that good student in primary school. I didn't like school. But then when I was 14, I read in a youth magazine that there's a new school for environmental studies, a new high school that opened in the Negev desert in Israel, in Sde Boker. It was new, but reading this article, I decided that this is where I want to go. This is where I belong. And I left home and moved by myself to Sde Boker at the age of 15 and studied in this environmental high school. It was fascinating. I loved it. And I'm involved ever since in environmental protection and zoology and science. So later on, I joined during my army service. I joined the Society for the Protection of Nature in Israel. So that the army is allowing some of the soldiers to work as the nature guides during their army service, and I was privileged to be chosen to be one of these nature guides. And then I just decided to go on in the same path and go study biology. And one of my professors was an environmental physiologist, the late Professor Amiram Skolnick, and his classes fascinated me because he answered all the questions that I always thought about, and we had some really interesting discussions. And without planning this, he actually asked me what my plans are after I graduate and offered me a master's degree, and this is how it started. So, and during all this time, I kept observing nature and asking questions, just like when I was a child. And some of the species I studied, I actually observed and learned when I was a soldier at the Society for the Protection of Nature in Israel. So in my army service, I was working around the Dead Sea in the Judean desert, and this is where the spiny mice that I studied later on thrive and coexist. There are two species, and the questions that I study actually emerge from observing these species and asking how come they can do things, or how come. So these two species, for example, one species is diurnal and the other is nocturnal, and they can switch activity patterns. And I was at the beginning, I just thought that it's interesting in perspective of shift workers, because they can shift their activity patterns, these two species, and then I got more and more into it. And when you study a species, you keep finding new stories and interesting stories, and now we are working in collaboration with many different laboratories around the world on these two species, on their depressed metabolism and skin regeneration and longevity and kidney physiology, and everything we touch is interesting. So I'm just following these questions from one question to the other.
Yeah, the spiny mice has always been, I always like looking at sort of these the species with the superpowers, and especially the regeneration dynamics as you were just talking about, because we don't
Uh-huh.
normally think about the rodents having that. We usually think about the amphibians and all that, but very
Mm-hmm.
interesting regeneration dynamics. Very interesting. You published a lot about the social dynamics of the animal, but clearly, the two major components of what you get into later on in your postdoc work, and after that is on the thermoregulation and this whole area of chronobiology, which I think I'd love to sort of we touched on it a little bit on past episodes, but would love to sort of start off again, almost like as if we hadn't. Here we have an area of chronobiology, sort of defined as the science of cycles, whether we're talking about lunar or solar or tidal cycles, all that have impact on biology. Would love to sort of get a sort of a 2024 lay of the land of about chronobiology from you, some of the I guess subset themes, because we hear about chronomics a lot now, and the ability to understand the genome and the proteome and all the neat magic that's happening below the surface. Chronotherapeutics, which is also was something we'll touch on when we get into our understanding of maybe taking something when the sun is out versus later in the day, and so forth, maybe more beneficial to humans. But let's start off with, if you wouldn't mind, sort of laying the base for where we are in sort of chronobiology 2024.
So I think that the main point is that all species on Earth developed in a cycling world. So as you said, we have the day-night cycle, the lunar cycle, and the yearly cycle, the annual cycle, and all species have to be adapted to these cycles. So there's the best time to perform a certain activity or function. So if we look at the seasonal cycle, for example, in order to be able to breed at the right time, for example, you have to give birth when the offsprings have the best chances to survive. And this happens normally in mammals, for example, during the beginning of the summer or the spring. But for the offspring to be born during spring, you have the females have to get pregnant ahead of spring. So they have to forecast when spring comes. And they do that by measuring day length. So day length changes along seasons. We have short day length during winter and long day length or light hours during summer. And by measuring the length of the day, actually what we're measuring is the length of the night, not the length of the day. But by measuring the length of the day or the night and the direction towards what it changes, so if it shortens or lengthens, we can actually estimate what's in what season we are in and towards what season we are advancing. And the same thing happens when you look at migration, at fat storage, at hibernation, everything that changes seasonally. And it's interesting that humans used to be very seasonal too. And there are studies even today showing that our immune system is adapted to different challenges during different parts of the year. Even reproduction is seasonal. The amplitude of this rhythm is reducing over the years since the Industrial Revolution, but it still exists. Even in humans, and it exists, of course, in animals and plants. And the same goes for daily activity and daily cycles. So, for example, cortisol is secreted before we wake up and prepares our body for activity. And if you wake up earlier, your blood parameters will be of a sleeping person, not of an awake person. And the same goes with your cognitive function, physiological function. Everything functions according to what is expected to happen during this time of day. Mm-hmm. So the main advantage, the main evolutionary advantage of and this is all controlled by a physiological mechanism within our body. In mammals, it's in the suprachiasmatic nucleus. It's two nuclei that are positioned above the optic nerves. And the main advantage is that it allows us to predict rather than respond to the environmental changes. So if we look, for example, if you're growing a cat or a dog, you know that they change their fur between winter and summer, and then again between summer and winter. So during winter, they need a thicker and more isolating fur. So, but they have to do it before they're exposed to the cold. If it's too cold already, it's too late to shed their summer fur and change it to a winter fur. So they actually have to predict the change of the season. And as I said, the same for the change of the days. And there are several studies that were published relatively recently that show that even that humans even respond to lunar cycles. For example, menstrual cycles of women apparently used to be synchronized to the lunar phase. And we see it in many other species, but it was a surprise when we found records that predict this in humans. And working on this on these cycles actually brought me again to environmental protection, because many human-induced changes actually change this periodicity. So, for example, if we look at climate change, the seasons are shifting. Summer is getting longer, and suddenly this day length is not such a good predictor for external conditions. And we see mismatches between the physiology of the animals, the timing of life history traits in animals, and the environmental conditions. So we can see, for example, birds that migrate too early or too late, amphibians that wake up before there are winter ponds, and many or birds that lay their eggs. As I said before, they have to lay their eggs so that when the sibling hatch, there's food for the siblings. But many of them eat worms, and sometimes the worms are already over when they hatch because there's a mismatch between. So the insects respond to temperature, but the birds have to predict when they will have abundant food, and this doesn't happen, and the chicks just die from starvation. So we see a lot of mismatches. And on the other hand, if we look at the use of artificial light, for example, so light is a very reliable cue. If we look at natural light, if it's dark, it could either be night or that you enter into a cave. But if there's light, it means it's a day. There's no false positive with strong levels of light in nature. But nowadays, when we use electricity, and after the invention of LED lights, which are very, very cheap to use, and the increase in electricity availability, we light huge areas, and most of the world is exposed to light during the night, and this changes the exposure to light, and the cue, the very reliable cue of light is again no longer reliable. So we see a lot of influences of the way that we change our environment on species ecology and health, and also on human health. So as you see, the issue of chronobiology and environmental protection actually I found joined again, and now I study a lot the influence of light pollution, for example, on the immune system of wild species, and on timing of mating and reproduction and fur changes, and we see a lot of adverse effects on wild species of our actions.
Yeah, it's interesting because even some of your more recent papers talk about and we'll get into this in a little bit, but even effects down to the level of the microbiome of various microbes, and sort of this whole One Health connection between human, animal, and the environment. And we'll go there in a little bit. One thing I'd love for you to touch on, and I really appreciate that introduction because it lays the groundwork for every place that I want to go into discussion. But a very interesting area that you point out in your work, we have nocturnal organisms, we have diurnal organisms, and in the biomedical research space, interestingly, we spend a lot more time on the nocturnal. I can't think of how many, whether it's the mice or other types of rodents that obviously study in the pharmaceutical industry and testing drugs initially. We cure a lot of those animals. Unfortunately, that doesn't always translate to humans. But clearly, we need to spend a lot more time on the diurnal species. We humans are diurnal, and hence, that it would make a little more sense. Talk a little bit about some, because I know you study anxiety in animals, depression, obviously the onset of diabetes, and this relates to the spiny mouse. Talk a little bit about some of what you've been spending time on here, because I think it's important that, in these major areas of unmet human medical need, that we focus our early research in discovery on the appropriate species. Talk a little bit about what you're learning here. I think this is very important.
So this is a really interesting question, because I don't think that many people know why we use mice, and previously we used a lot of rats as our key species for biomedical studies. And the answer is really simple. Because it exists around us, it was easy to trap a mouse in the lab, in the field, or at home. They breed really, really well. They're small. They eat everything. And that's why we use them. There's no scientific reason why we chose to study mice as a model for humans or for mammals in general. And there are many different questions regarding this choice and how it affects the progression of science. But the issue of activity pattern is really interesting and important. So just to start with, if we come to the lab during the day and study the behavior of our mice or the physiology of our mice, we actually work on sleeping animals because they sleep during the day and they're active during the night. So unless you shift the light-dark cycle of your animal house and work on them in the dark, because if you turn on the light, again, it's an intervention which affects their behavior and physiology. So unless you do that, you work during on a reverse light-dark cycle, and you work during the night, you're actually working on sleeping individuals. So this is just a practical issue, but the more interesting point is that being nocturnal is not simply the opposite of being diurnal. So you can't just take the results you have and mirror them to nocturnal or diurnal activity. The ancestral state of all mammals was nocturnal. And nocturnal species, if you compare them, especially their chronobiological system, their circadian clock, it's very conserved. But diurnality actually evolved in many different lineages and in unrelated occasions. So it makes sense that diurnal species are not all the same. So this is one issue. So even if we work on a diurnal species, it doesn't mean that it's the same as another diurnal species. One interesting point, for example, is that melatonin, which is a hormone that is secreted from the pineal gland, is secreted only during the night, both in nocturnal and in diurnal species. So the circadian clock determines that the melatonin will be secreted during the night. And if you turn on the lights or you're exposed to light, the secretion stops. But if there's dark during the day, melatonin doesn't start. So if you look at a nocturnal species, or let's start with us, we take melatonin when we want to go to sleep because it's secreted during the night, and it means that during the night we have to go to sleep. So it signals our physiology, the physiological systems, that it's time to go to sleep. It lowers our body temperature, our heart rate, our blood pressure, and we feel sleepy. But in nocturnal species, it's opposite. Because when melatonin secretion increases, it means that it is time to wake up and go out and forage. And this is this is completely opposite, and we don't know what the difference is. So because everyone is working on a nocturnal species, we actually don't know how melatonin affects function in diurnal species, including us. And this is a very, very, very basic question. How, if you look at the biological clock at the suprachiasmatic nucleus, gene expression, electrical activity, everything is the same. The clock shows the same time in nocturnal and diurnal species. It affects the pineal gland to secrete melatonin in the same way. But melatonin, which transfers the information from the central clock to the body, actually conveys the opposite information. In one species, it says time to wake up and start being active, and the other species, it's time to go to sleep. And we really don't understand what the difference is, how this difference evolved, and what is the biochemical mechanism, which is amazing. I think it's an amazing question. And then if we, for example, look at seasonal affective disorder in humans, which is a depression, reoccurring depression that develops during short days during winter, and there's a remission, a spontaneous remission during summer. If you take a nocturnal mouse and you put it under short photoperiod, it actually has more time to be active. And actually, if you look at the mouse in nature, during daytime it's in its burrow in the dark, and during nighttime it's out and active. So having more dark hours is actually more similar to the natural condition. So if any effect is expected, it's in the opposite way. So having short days should actually be better for him in the lab, because in the lab he's exposed, that they're exposed to light, which in nature they're not. So for example, to test this very, for me, straightforward question, we just took day active species and tried to expose them to short photoperiod and long photoperiod, and showed that there's a similar effect to what we see in humans. And then we tried to connect it to melatonin effect. So during long nights, we have more melatonin. So we tried to imitate the long nights by injecting the mice with melatonin instead, the diurnal mice with melatonin, instead of exposing them to long dark hours, and we got the same response. And I'm not expecting that this will happen in mice. And this is a relatively straightforward example, but there are many other examples where we're not using the optimal animal model for our studies in many cases. And I think that we're just using mice as a mammalian animal model without asking which species is the best model to study the question I'm interested in. And one of the reasons is that it's really easy to hold and breed these mice. And also that there's a lot of literature about them. So we don't know as much as we know about mice on any other species. When you publish a paper about some strange spiny mouse, the chances that mice people will cite you is much lower than if you publish a paper on lab mice. So it's also more difficult for the scientists. And another interesting question is our lab mice are as similar to each other inbred as possible, which is on one hand a good thing, because we can find differences in relatively small sample size. There's no much variation between individuals. So if you want to test an effect of a drug, for example, you can use 5 or 6 mice, sometimes 3 mice, and the response is very, very clear, because there are almost an exact copy of each other. But if you take a wild type animal, there are huge differences between individuals, just like in humans. You're not expecting to get exactly the same response from each human you're treating. So the history has an effect, and the weight and the height and your social contact and your emotional state, and we're different from each other. And in mice, it's not the case. So in some cases, it's good because you don't need to use a lot of animals to get a significant response, and you can understand the mechanism. But but if you want to compare it to humans, sometimes we show very different responses between males and females, between different origins, people that come from different countries, between people that consume different diets, and we don't see it in science. So I have a lot of thoughts and questions about the use of mice. I understand why people prefer to use mice. Their genome is sequenced. You don't have to validate the methods you're using. Everything is ready made. You have protocols, you have kits. Everything is really easy. And if you want to develop all these things for a new species, it's a lot of work, and you don't get the same reward in publication and as you do with mice. But I think it's more interesting.
I'm thinking, and it's sort of a separate show, I guess, but melatonin never works for me. And I'm just wondering if there's probably something chronogenomically different, but whatever. We'll go there some other time. But talking about the importance of working on these, lesser worked on species. You clearly, obviously, the spiny mice is somewhat related, but when you get into your work now with thermoregulation, sort of the breadth of the organisms here are much wider in the sense that, and we've talked a little bit about torpor on previous shows, but more from organisms hang out in very cold places. So bears, we talked about the Arctic ground squirrel. And again, coming at sort of the angle of slowing metabolic rate and energy consumption to not losing, the ability to have heart health in the case of the bear, or the Arctic ground squirrel's brain, collapses in many ways, but is preserved. When you get to some of these desert organisms, I'm very interested in sort of I'm personally interested in sort of the difference between animals that enter a state of torpor in very cold environments, very, very, very hot ones like in the desert, but also, some of the things, again, because you were mentioning about, the way you study in the chronobiology space in terms of melatonin and light exposure and all that. You have a series of papers here on other very interesting sort of interventions. There's one from 2009 on something called melanin concentrating hormone, and the ability to affect energy expenditure and body temperatures in mice. I'd love to sort of also get your perspective on sort of some of the intervention angle there as well, because clearly in learning about torpor, and ultimately for the human experience, how we can mimic, again, some of these superpowers, not necessarily to hibernate, but to preserve health in our various body systems, as these organisms do, I think is a really cool area of exploration. Love to hear your perspective on all that.
So the cool thing about the desert animals that enter torpor is that they do that in a relatively high ambient temperature. So one of the interesting things about species from cold habitat, like not the bears, but the ground squirrels or bats, insectivorous bats, they actually lower their body temperature to even below zero, to -2, I think that even -3 degrees Celsius. And in this temperature, the question is how to protect your body from freezing, but the rate of biochemical reactions is so slow at these temperatures that energy expenditure reduces just because of the reduction in temperature. And in desert animals, this is not the case because, for example, the harsh season could be the summer season and not the winter season. So temperatures could be as high as 30, 35 degrees even during the night. So actually, we don't see a very large reduction in body temperature when they enter torpor. But when we measure their energy expenditure, it can easily reduce by half. And one of the things we're interested in is how they do that. So this is not temperature effect. It should be some sort of a molecule or a biochemical mechanism that suspends energy expenditure. And the truth is that we still don't know what the answer to this question is, but I think this it's an amazing area for research. We tried working on it a bit also with collaboration in collaboration with the universities in the States, but we still don't have an answer. And this is something that we can certainly learn because if you look at humans, if you want to use it in humans, then lowering body temperature is actually inducing a stress response in humans because your body temperature reduces and your brain starts sending signals to the body to start producing heat, shivering and you feel bad. But if you can reduce metabolism without reducing body temperature, this is a completely different thing. And this can be used for example, for shipping organs, for implantation. If you can reduce their metabolism, it can be used for an injection when you have people that are wounded and losing blood.
Yeah.
If you can reduce their metabolism, their oxygen consumption, then you can extend their life, the time that they still live under these conditions, and increase the chances of rescuing them. So I think that this is a really interesting question. There's an interesting issue here that I'm not sure it's true, but I keep thinking about it. That many of these desert animals live in the Mediterranean and Africa and maybe in Australia, but most research developed in cold countries in the United States and Europe. So I think that maybe the desert species received less attention than species that live in cold areas and in snowy areas. But I'm just guessing. It's just a thought. I don't know. Never checked that, but but I think that there's a lot of potential in studying these desert species, not only in terms of lowering metabolism, but also in terms of water economy and being able. So spiny mice, for example, in nature, they don't drink at all, and they have a very high ability to produce highly concentrated urine, and we also have now a study on their kidney function and to see if they have special abilities or mechanisms to increase urine concentration and reduce water consumption. So, but I actually think that spiny mice are not that special. It's just because I started studying them, because I observed them. And when you study a species, you learned about its abilities. And I think that many species have very unique abilities that we don't study and we don't know about. I don't think that spiny mice are special in this term. Actually, every species that you study, you find interesting things that you can follow in a research. So.
There's a lot of them out there. And the fact that we're only one of 9 million on the planet, I think that's, there's a lot to branch out to. No, but that's, I appreciate it, especially when, you were mentioning that sort of that golden hour concept of saving somebody sort of in a trauma situation and all that. I think thermoregulation and this whole area of torpor is just, we've only begun to touch the surface. So I'm excited that you and other important groups are working on it. So that's really great. Um, so I could segue a bit to, some of the environmental level topics now. But before we do that, I would love if you could just sort of introduce us, because, I mentioned in your bio, you wear both of these hats, not only at Tel Aviv University, but at the Ministry of Environmental Protection. Could you just talk about the role, a little bit about how you were elected to this role, and clearly, um, you know, when you're working on chronobiology and thermoregulation and other interesting things you're doing, but the minister calls you up over here and says, Professor, we have a situation in the lower Jordan River where there's too many hormones from being in the effluent. I just pulled that out of one of your papers, but talk a little bit of how you balance these roles, because clearly, I mean, they're connected, of course, but they
Yeah.
could be quite a diverse set of problems and issues that you have responsibility for per the environmental, scientific, and technological purview of the Office of the Chief Scientist. So walk us through a little bit about your responsibilities there first, if you would.
Yeah. So during all the time that I was a faculty member at Tel Aviv University, I always spent some time in nature conservation, as you said, in the Nature Reserve Authority and the Society for the Protection of Nature in Israel. So I always spent about 20% of my time trying to improve the environmental situation in Israel, protect the environment and wildlife. And as you said, I was the chair of the School of Zoology, and I'm a full professor. And I started to think what my next role should be and how I can influence, make things better for the environment in Israel. And I just ran into an ad at the newspaper saying that the ministry is looking for a chief scientist. So I applied and got the job. And I'm actually on a leave, or part-time leave from the university. So I'm working 50% time at the university and spend most of my time with the ministry. And I see my role as connecting between the academy in Israel and the work of the ministry. So I'm trying to improve the work of the ministry by providing them with the most updated academic knowledge. Mm-hmm. So it's not always me that have the knowledge. I just connect between professors from around Israel that I met and I know over the years. I lead several projects, for example, with the Israeli Academy of Arts and Sciences, that help the ministry to do its job better. And the Academy is really happy to volunteer and help. Most people are very enthusiastic about the opportunity to come and support the work of the ministry. Mm-hmm. And yes, sometimes the questions that I have to deal with are very far from my expertise. So hormones in the rivers are relatively close to what I do, but sometimes I work on a completely different questions and projects. But it's amazing because you can actually influence the state of nature in Israel by being at the ministry. And when I joined the ministry, I made a list of things that I want to work on. Eventually, I think that I didn't deal with most of them. Because when you get there, you understand that things don't work that way. I could go and try to push the things that I wanted to advance, but you have to look at the opportunities to advance things. And I just try to follow the office work and see how I can advance it. So, for example, now we're doing a huge project on using information from satellite to map environmental issues. And I never worked with remote sensing or with satellite information, but I realized that that we may be able to use information from satellites to improve the work that we do at the ministry. For example, now during the war in Israel, we have a problem with asbestos roofs that are being bombed or burned. And the ministry wanted to map all the asbestos both in the north and in the south, and clear it before it's burned or damaged, because then it's much riskier. It's a health hazard. And if you remove it before, it's much easier. It costs less. But we didn't want to send people there to look for asbestos and then risk them. So what we're doing now is, together with the Israeli Space Agency, we are developing a way to map using multispectral imaging to map all the asbestos in Israel, and then prioritize which roofs should be removed first and which should be removed later. So this is something that I never worked on, but I heard the seminar once, and I thought maybe I can contact this professor and ask him if he can help us. And then I approached the space agency, and together we created the method that will now change the whole way that we deal with asbestos, not only during the war, but also during normal times. So sometimes these are topics that, just common sense and some acquaintance with the Israeli Academy, and I can make the connections and advance the work. So
it's an amazing suite of responsibilities. But thinking about the satellites for a minute, and obviously that, in addition to what you were just mentioning, obviously allows you to look at as you were mentioning earlier in the show, the light pollution issue. And then, I guess trickling down from there, because you have been, focusing on a lot of work per this eternal summer that we've artificially created, and then of course, as you were mentioning, the trickle down to the ecology, the environment, the organisms, and one place that your research has gone a little bit here is to the microbes, not just in certain microbial communities and the microbiome, but also, as you're again, there's this one interesting paper you wrote on sort of the drivers of infectious disease seasonality and potential connection to COVID. Again, we wake up a certain species too early; it doesn't eat these insects, which would normally the bats would eat. So the whole One Health thing comes into play here as well. And I'd just love to, get your top line there, because obviously we think of all us, our megafauna and all that, but there's a lot of microbes and viruses and other things on this planet that are also affected by these cycles. And I would just, love to get your top line there if you would.
So I think that the way to look at it is it's like an orchestra. So when everything happens at the right time, there's a harmony. Everything works well, just like in a concert. But when we start shifting things in the orchestra, if the violin plays at the wrong time and the drummer plays at the wrong time, then there's no harmony and things doesn't work. We don't want to listen to it. And I think that in chronobiology, it's pretty much the same, because again, you look at things that have to happen at a certain time to work in harmony. Mm-hmm. And it's easy for us to look at the bigger species. You see them and you can relate to them, and the methods are relatively easy. But the same thing happens with microbes. and it starts from the viruses and the microbes, as you said, and then it climbs up. So if, for example, if we look at mosquitoes that transfer a disease, dengue virus, for example, they're active at a certain time, and we know how and for example, in Israel, we used to have mosquitoes that were active only during the night. So we could use, I don't know the name in English, window nets, or
Yep.
you could find solutions. But now we have also insects that are active during the day, and they don't have the right predators. This is an invasive species, and they are able to transfer diseases. So when you change nature and things don't happen at the right time, there are consequences. And I think that this One Health approach is looking at us as one species of a whole ecosystem. And when we change this ecosystem, eventually we damage it, and at a certain time it will get to us too. So I think that we have to think more at a more holistic way and see how we affect nature. And in many times, we can't predict what the consequences will be. So, for example, if you look at light pollution, one of the consequences of using street lights is that moths are attracted to these street lights. Sometimes they go around these street lights until they die. Sometimes insectivorous bats take advantage of the fact that they are near these street lights and they prey on them. But the result is that especially large moths are disappearing. And many of the large moths don't exist anymore. And you could say that's not bad, but moths are very important pollinators for some crops. So you're damaging pollination. So I think that there's a cascade of ecological consequences that we don't think about, and we have to study and learn and protect nature in general, because we can't. These are very complex systems, and we can't always predict what the consequences will be.
Absolutely. And this loops back to one other topic I'd love to just touch on with you, and I really do appreciate your time today, Noga. I noticed this was back in 2010, you did some publications on sirtuins and the connection in that case to diet-induced obesity, but broadly, obviously, this area has broader implications in the whole area of health span and aging. And I found it interesting. Your most recent paper, which just came out the other day, this was in Diabetes Metabolism Research and Reviews, talking about this overall principle of circadian syndrome, a little broader than metabolic syndrome, and things like cognition impairment in adults. And I'd love to, again, just get your top line here, because I know this was a rather multidisciplinary sort of international group with you and Saudi and Finland and Australia and a bunch of researchers from all over the place. Broad sort of thoughts on circadian syndrome, aging, and this evolving area. I guess we'll call it health span biology, which is kind of embryological right now, but really coming online as a potential interesting sort of therapeutic segment in its own right.
So this actually started in a discussion over lunch in a meeting with Paul Zimet from Australia, who actually published the work. He was one of the pioneers in metabolic syndrome, and we talked about the metabolic syndrome, and I told him that I think that the circadian system is highly important in the development of both obesity and diabetes, and I mentioned that I already had back then some studies on depression, and the studies on depression I did them on sand rats, which are diurnally active, and Paul actually did some studies on sand rats on diabetes in the early 60s, I think. And I told him that I think that it could be that the reason that these sand rats develop diabetes is because they are diurnal species, and we bring them to the lab. And when we bring them to the lab, they lose their rhythmicity. And I think that the fact that they are eating during both day and night, and their whole circadian system is messed up in the lab, that this is the reason why they develop diabetes. So he wasn't working on the reason for the development of diabetes, but on the mechanisms and the consequences. And the sand rats are known animal model for diabetes because they develop diabetes spontaneously when they're transferred to the lab. So I told him, I think this is because they're diurnal and they lose their rhythmicity when they're transferred to the lab. And we started working on that. And we did some studies on sand rats, trying to manipulate their activity pattern and their activity levels, providing them with running wheels, changing light intensities. And we found that we can actually influence the development of type 2 diabetes just by changing environmental conditions, which was amazing. He didn't believe it first. And just by changing light levels or providing them with a running wheel, keeping them outdoors when they have signals from other species and natural light, and if I keep them in the same cage outdoors, they don't develop diabetes. Same food, same everything, same cage, just in a shade where they're exposed to natural environment, which everything in the environment cycles. So it's not only light levels, it's also temperature and smell and noise and the predators, and everything changes. They're not exposed to the predators. They can't be eaten, but they sense them. And they don't develop diabetes. And this is how it all started. So we started working on other diurnal species to see if they also change their activity patterns, because we already knew that the golden spiny mice and the sand rats change their activity patterns. So we started working in collaboration with many different labs on other species, on other diurnal species, and we found that the same happens. Most diurnal species, even if they're not studied, just kept in zoos, they in the holding instructions, they always write that you have to limit their food consumption, because otherwise they develop diabetes, all rodents. Right. So I think that except squirrels, all diurnal species, rodent species that we studied, lose their rhythmicity and develop diabetes under laboratory conditions. And then, the group that worked on this project started growing, and we decided to look at human databases. And my studies showed that they also develop depressive-like behavior. So we wanted to look also on cognitive function and depressive-like behavior. And actually, when we work on human databases, we find a very similar thing. So if you include circadian system-related parameters like shift working or problems falling asleep or unstable sleep patterns, the incidence of diabetes, obesity, CVD, and depression increases. And it's actually a better predictor of the risk of developing these diseases than just the metabolic syndrome. Yeah. So I think that especially in the modern society, when many of us work shift, we sleep late and wake up early. We don't have enough sleep hours. We can eat whenever we want. There's no order in eating, and all the fashion of restricted feeding time that people are using now is actually based on circadian rhythms and the right time to eat, and that there's time to eat and time to avoid eating, and it's important for our body function. And many people are actually not exposed to dark during the night. And when you're not exposed to dark during the night, you don't secrete melatonin. As I said at the beginning, melatonin is secreted during the night, but if you're exposed to light, the secretion stops. And the wavelength that influences melatonin secretion most is the blue light. And most of our screens emit blue light. So you have the option now in many of the phones to choose a night mode, which reduces the blue component of the wavelength of the light. But still, you're exposed. If you're reading from your phone or from an iPad, you're actually exposing your eyes to light during the night, and it reduces the melatonin secretion. So I think that this circadian medicine is a very important issue that doesn't get enough attention as it should, especially when people are trying to live a healthier life. Part of healthier life is having a good night's sleep, being exposed to light during the day. Light is not bad. We have to be exposed to light during the day. So go out at least once a day, being exposed to sunlight, and then make sure that you're in the dark during the night. And many of our sleeping rooms are not dark during the night because there are lights from the windows and from electronic things that we keep, like a phone or a TV or in our bedroom, and this is bad for our health.
So obviously, obviously there's simple adjustments that we can make there that make all the difference. But at the same time, as you're pointing out, these are areas that I'm in full agreement with you are going to be critical as we continue to dive further into all of these unmet medical health conditions that are responsible for the trillions of dollars that we spend on health every year, and really sort of completing the story. Let's put it that way. So I'm really happy that you're focused on it, and hopefully we can, not just the chronobiology, but the whole area of thermoregulation, and hopefully you can raise awareness there and make further connections. But really, it's such an impressive story, Noga, and set of responsibilities, and really wishing you and your team the best, or both teams, both the Tel Aviv University and the ministry, with all these programs, because they're just so very critical. And it's great hearing about them, and really, I look forward to continue to follow what you're doing, and again, wishing you the best with all of it as you move these programs forward. Again, for everybody that's going to be listening to this particular episode of our show across the podcast networks or watching on the YouTube channel. Again, you've been spending time with Professor Noga Kronfeld-Schor, Chief Scientist, Israeli Ministry of Environmental Protection, Chair, Principal Investigator in the Department of Zoology at Tel Aviv University. Noga, I really want to thank you for taking the time out of your schedule to talk to us and educate us on all these topics. Obviously, thank you for everything that you do. And as we like to say on our show, thanks for helping to create a better tomorrow for so many people out there via these programs. Really impressive. And I look forward to staying in touch.
Thank you very much.
Great having you.